So our project is displayed on this poster, which studied Japanese nutweed and related plants in Wisconsin. Japanese nutweed and other related species are a part of the genus Reynoutria. This genus has had many different names in the past couple years and is currently classified as Fallopian japonica, which phylogenetic evidence supports. This is a smaller genus containing only around 5 species and is native to Eastern Asia. The green countries on the map represent the native range of the whole genus. Some of the species have very specific native origins, such as Sakhalinensis, which is native to Sakhalin Island right above Japan. The Japanese nutweed is of course native to Japan. This map supports the idea that this plant has a fairly narrow native range and a rather extensive invasive range, as we will see later on in this presentation. So the invasive Reynoutria species are a few species that are invading Europe and North America. They've caused problems where they're not native and have been active targets of control and study to try to identify them, leading to the motivation of this project, which was trying to learn a bit more about identifying these plants and maybe developing some tools for identifying them and potentially exploring the possibility that there are extensive hybrids between these two species. In the photos, you can see 2 pressed specimens at the same scale. Giant nutweed is capable of making really big leaves. Besides their large size, one of the features that helps identify giant nutweed are these big lobes at the base of the giant leaves. There are lobes in both giant nutweed and Japanese nutweed. The bases of the leaf blades are truncates, so they're just straight across at the bottom, which is one of the more useful features that can be used to identify the species. Looking at potential hybrids between the two species, there's a hybrid called Bohemian nutweed Reynoutria bohemica. This hybrid has intermediate morphological characteristics. So this is a plant that is very problematic. It grows very large and can grow using vegetative reproduction to fill a small area and compete against other plants. It also has a chemical tool known as allelopathy that one nutweed plant can actually kill its neighboring plants as a form of competition. Due to this reason and the speed of its seed production, it's able to spread pretty far and wide, and as with the case with many other invasive plants, this is a plant that for a while before it was classified as invasive, and even still today, people are moving it around intentionally due to its attractiveness, which can obviously be very problematic.
We primarily worked in Wisconsin in conjunction with the Department of Natural Resources. So we are looking at the genetic diversity of a species of knotweed in our state. So the picture on the right here is a picture of Wisconsin's diversity. And we might see something very different if we look somewhere else, such as Europe, where they also have an invasive knotweed problem, which is something that we're interested in given the interesting results that we have found thus far. So the background shows the reported ranges of 2 parent species. This includes the giant knotweed, which is in the orangish color. You can see that it's distributed greatly to the north. And then the blue color is the Japanese knotweed, which is distributed primarily in the southeast part of the state, and then is also distributed throughout the state as well. So the hybrids have been found outside of Wisconsin, mainly into Minnesota, where you can see in the green there. So we aim to do some DNA investigations using samples from across the state to see what kinds of genetic diversity there was and what kind of molecular markers they would be able to find among the different parent species and their hybrids. So we collected samples and looked at the morphology as a method to identify them in a more traditional sense. And we also looked at their DNA data. As far as getting the DNA data, we used a couple of genes that have been used before that's standard to something that we have known to be useful in this group. So we were getting signals from the chloroplast using the MATK gene from the tools we were using, and also from the nuclear region and the nuclear chromosomes using the leafy gene. So primers have been developed for the leafy gene in knotweeds and other members of the family that have been investigated using the leafy gene, specifically the second intron. So introns offer more variability and more evidence for unique sequence combinations in individuals, so this will help us to identify the different species. This is an example for the alignment on the bottom here that we got for the leafy gene. So you can see some locations where there are nucleotide substitutions and some places where there's also gaps. This was the basis for our phylogenies that we will get to in a couple slides. So we use traditional primers that have been developed. We determined that there are multiple sequences from single individuals, which sometimes happens as a result of hybridization or polyploidy. So we had to develop primers that were specific to the Japanese knotweed types of sequence and the giant knotweed types of sequence that allowed us to get a clean version of the different gene sequences from each individual.
Taking a look at the result of our phylogenies, this is the MATK gene, which is the chloroplast gene. The chloroplast genes are inherited from only one parent, and there's typically only one kind of chloroplast per individual. It is usually a pretty strong indicator of whether the plant is either a Japanese knotweed or a giant knotweed. This does not clearly identify the plant, but it at least shows what kind of plant the female parent was. The background sequences that are not highlighted were found by other researchers, so our sequences are following those that have been reported by others. Curiously, though, our sequences for the giant knotweed have yet to be reported in North America. The more complicated picture comes when you look into the leafy gene. The leafy gene is a nuclear gene, and this plant is known to be found as tetraploid, which means it has 4 chromosome copies, but it can also have other ploidy levels. So it is possible for these plants to be found with 4 or 6 copies. With each copy of the chromosome, there is the potential to get a different leafy sequence. Luckily, you can identify the type of sequence by itself and determine if the individual has more than one sequence present. Some of the individuals we had only had one leafy sequence, which is a tad easier to determine. It is more common, however, for an individual to have both a giant and Japanese knotweed sequence, and this is represented by the spider web of connecting lines. This is a good signal that you have a hybrid, and this is how we interpreted it in most cases. We are also seeing evidence of different types of hybridization. Not only is it hybrids between 2 pure parents, but also hybrids between hybrids, and then also hybrids between hybrid and pure parents.
So we aren't 100% sure on how the genetic combinations came about, but this is one way to think about how they might have originated with some potential parent individuals and the genotypes that we observe. So everything with a box around it is a genotype that we found in our study as compared to the ones without boxes, which are essentially pure versions of the species that may have existed in an individual, or perhaps they're polyploid individuals, that there may have been starting individuals with 2 combinations of the sequence already. But we see lots of combinations of hybrids, so there's no clear pattern as far as J1 and J2 and S1 and S2 and so forth. Almost every combination is present among these different leafy genotypes, which is why we might have to investigate other markers or work out the finer details, such as source populations, to sort of delineate the parental species, their original location, and when they were first introduced. So now, referring back to the geography of Wisconsin, we see, similar to what was mentioned at the beginning, that there are little more plants identified as Japanese knotweed in the southeast through the DNA for the chloroplast, which is the upper part of the pie charts. And individuals that came in with the giant knotweed chloroplast sequences, shown in the north part of Wisconsin, 24 and 37, the bottom half of the pie chart shows the leafy contribution, showing that most of the individuals that we sampled would come out as hybrids. So one form in the blue shades, which is the Japanese knotweed, and one form in the brown and orange shades, which is giant knotweed. And it appears that the majority of these have a combination of Japanese and giant knotweed elements in their nuclear DNA. And this helps to explain a little bit why we were having trouble identifying these using their morphology alone, and really opens up a lot of uncertainty about what we would find if we looked in other parts of North America, if we got the same kind of sequencing from Europe and so on. And we believe that we have made an interesting contribution to knowing what is out there as far as genetic diversity in this group of invasive species, and have made a lot of room for future investigations.